Cell rest. You should be super comfortable now that photosynthesis makes the macromolecules or the precursors of them, and then cell rest takes those macromolecules and reverses the process to pull out the high energy electrons, ultimately to synthesize ATP. Okay, so this is a synthesizing reaction and then a digestive reaction. All right, the whole point is energy in from the sun and in the form of light, and chemical energy out. Okay, so this process of synthesizing all of this ATP, there's 3 major steps to it. Some of your college professors will teach it as 4. I teach it as 3 because that's my training, and it also matches what IB does. So there's glycolysis, which occurs in the cytoplasm. Then there's the Krebs cycle that happens in the core of the mitochondrion, what's called the matrix. And then there's the electron transport chain, steps 1, 2, and 3. The reason why some people teach this as 4 steps is there's a reaction here that converts the pyruvate into a compound that can enter the Krebs cycle. And that's commonly called the link reaction. And a lot of us combine the link reaction with Krebs. However, some of your professors will split this out and they'll say there's 4. Fine, whatever they want, give it to them. Okay. And then there's a shuttle here by a protein called malate. And some people account for the malate shuttle and some people don't. And so you'll hear different amounts of ATP being produced total. I was trained 36, and so that's what I teach. IB does the same thing. But some of your professors will say 32, 34, as high as 38 if they ignore all the shuttles. And so it's not that I'm wrong or they're right or I'm right. It's how specific do you decide to get on the biochemistry. Ultimately, whatever the professor asks for, give it to them. It's real simple. It's their class, right? Then we started to get a bigger picture idea, because ultimately we are ripping out electrons. I need to close a door and I'll be right back. I'm out of here. I actually had my biochemistry professor do that, right, my junior year. Have I ever told you guys that about my first semester of biochemistry, how our professor quit at the beginning of the semester, about the 3rd weekend? Have I ever told you that story? No, we were sitting there. We're in a lecture hall of about 150, 200 people. And he's in, the middle of lecture, and all of a sudden he pauses, and he put the markers down. He looked at us and he said, "You know what? None of this really matters." And he walked out the door. Right, seriously, and we all just sat there for a minute or so waiting for him to come back. He didn't come back. It was really weird. Still is. And pretty much we picked up our stuff and walked out. Oh, that was on a Tuesday. We showed up Thursday, and the dean of the chemistry department came in and said, "Your professor resigned on Tuesday." And what he basically did is he walked from our lecture. Not that we were bad people. He just decided to quit. And he walked over to the dean's office and said, "I quit. Here's my keys." And he quit his job on the spot. So the dean told us, "You will have a new professor on Tuesday." And we're like, "Okay." And so they actually recalled a professor who had retired. And okay, you take your quintessential grandma, right, with the gold rimmed glasses and gray hair, and that's what walked in the door on the following Tuesday, right? So we're all 20, 21, 22, right? And here, seriously, this older woman, this grandma, comes walking through the door, and we're like, "What's grandma gonna know about biochemistry?" Grandma had her PhD and had been teaching longer than any of us had even been alive, right? Grandma knew her stuff. She was a retired professor. She was fantastic. And so we lovingly called her Dr. Grandma, and she was awesome. But then when I got up and left right there, I had a flash of when my professor was in the middle of class and he quit. Seriously, he walked out. It's amazing. You guys will have similar experiences. The more people you meet, it just happens. All right. So this can look like a complicated diagram. It's not. All right. There's 3 major steps or phases to cell rest. Okay. There's glycolysis in the cytoplasm. There's Krebs in the core of the mitochondrion in the matrix, And then there's the electron transport chain, where there are protons that are pumped and a proton gradient and chemiosmosis and all the same chemistry that we talked about in photosynthesis. Since you're ripping high energy electrons out of substrates, all right, you're ripping high energy electrons out of macromolecules, there is a tremendous amount of electron transport. That's what these are. Okay, one carrier is very, very similar to what is in photosynthesis. The electron carrier in photosynthesis is NADP, all right? And the electron carrier in cell resp is NAD, same job, all right? There is an additional electron carrier in cell resp. It's a flavonoid, FAD, and the only difference here is that when it picks up its electrons, it picks up a little bit more than NAD and actually becomes FADH2, right? But all that's happening here is the high energy electrons are getting ripped out here and they're being transported here. And you know why? Because when the electrons flow through or are passed through, I should say, the electron transport chain, that energy is then used to create a proton gradient. Once you have proton gradient, you can then have chemiosmosis. Once you have chemiosmosis through an enzyme, you can phosphorylate ADP into ATP. Chemistry is the same, right? And then at the end of the ETC, you have these low energy electrons, got to get rid of them. So you bond them to protons and oxygen to make water, okay? And don't forget that all eukaryotes do this. So plants do this same process. So plants need oxygen as well. Comfortable? You okay?
Yep.
All right. So we just need to go step by step. So these three boxes, 1, 2, 3, and then these three steps, 1, 2, 3, are simply in this diagram. Ready? 1, 2, 3. Okay. It's like you're doing the, the cha-cha here. 1, 2, 3, 1, 2, 3. Because you guys know a lot about ballroom dancing, right? Do any of you know how to ballroom dance? Do you guys know how to waltz or tango?
Not at all.
Not at all. When I was a kid, my great grandmother on my dad's side was a piano teacher in Iowa, of all places, and she used to play piano in the movie theaters for silent movies. Okay, and so to say she was really into music is an understatement. So when I was a kid, she paid for me to go to ballroom dancing classes. Seriously, I was like 7, 8 years old doing the tango. It's hilarious. I took it really seriously too. Of course I did. Grandma was paying for it. But I will sometimes mention, ballroom dancing, and people just look at me and I go, "Oh, most people don't know how to ballroom dance." It's really, really fun. Especially the tango, man. When you do the tango, you just get to dance with attitude. It's actually encouraged. It's really fun. To mention when you're a kid, it's a cool way to meet chicks. Just saying. All right, so which organelle is this here? What is this kidney bean thing here? What is that?
Mitochondria.
That is the mitochondria. Always remembering where we are in the cell. So if this is the mitochondrion, then what is this here? What is this fluid here?
Cytoplasm.
Yep, cytoplasm. Okay, and then I toss the plasma membrane up here. Now, if this is a plant cell, of course, we'd have the cell wall up here, but this could be an animal cell. This could be a fungus cell. This could be a protozoan, right? This could be any eukaryote, because eukaryotes use mitochondrion to make ATP. Bacteria, they actually have ATP synthase embedded in their plasma membrane. That's a whole nother discussion. All right, so step 1, step 2, step 3, what goes here?
Glycolysis.
Glycolysis. Glyco meaning sugar, lysis meaning split. This literally splits sugar. Takes any of the rings of sugar, whether it's a 5-carbon sugar, 6-carbon sugar, doesn't matter. Glycolysis splits it in half. What it really splits it down into is this molecule called G3P, if you've ever heard of it. It's really what glycolysis does. It takes all sugars back down to G3P. Okay. So, which sugar did we put here? Always, it's the gold standard for biology when you teach this stuff.
Glucose.
Yep, this would be glucose. How many carbons?
6.
6 carbons. Boom. Good old C6H12O6. Okay. All right. So you take this 6 carbon chain and you cut it in half, right? So if you take this 6 carbon, shouldn't say chain, 6 carbon ring, right? And you cut that ring in half, you have 2 chains. So does it make sense that you have 2 compounds over here? You haven't created anything, right? Because you still have the same 6 carbons. They're simply cut into 2 3-carbon pieces. Does that make sense? We're not making anything, right? Nothing new. There's no CO2 in. All right. And what's the fancy name of this 3-carbon compound?
Pyruvate.
Pyruvate. Some of your professors will refer to that as pyruvic acid because in water this ionizes to its acidic form. Fine. All right. Whether they call it pyruvate or pyruvic acid, same thing. The star. What does the star always mean in any biological diagram?
ATP.
Always. ATP. This process makes 2 of them by a process called substrate level phosphorylation. There's no ATP synthase, there's no chemiosmosis, there's no proton gradient here at all. The phosphate is directly attached to the substrate, which would be ADP, through a process called substrate level phosphorylation. It's not something we're going to talk about here. You'll get it later. Okay. All right. So then you look at step 2, I'm going to ignore that. So what is step 3 over here? Because that will help explain what this arrow is. What's step 3 over here?
The electron transport chain.
Yep, the electron transport chain. Now, you will hear some people talk about the electron transport chain, and you will hear other people talk about the electron transport system. Okay. All right. Technically, the chain is just the proteins that handle the electrons. That's just the electron transport chain. If you include ATP synthase in with the chain, it's called the electron transport system. Okay, so if you hear someone or a handout or diagram or some animation refer to the electron transport system, that's just the chain plus ATP synthase. Okay, electron transport system isn't a term I use very often because to me it's just one more term that you have to fit into your head. So I really try to stick to electron transport chain, but I have to mention system in case you hear it. Cool. So all of this arrow here and this arrow here are the high energy electrons that are being delivered to the electron transport chain. Okay, so that means that there's an electron carrier here, there's an electron carrier here, and in just 1 second we will add an electron carrier here for the link reaction. Fine. So which electron carrier here carries high energy electrons from glycolysis over to the electron transport chain? Which carrier goes right there? NAD with the electrons would be
P.
Mm-mm.
Oh, NADH.
NADH, yep. Okay, it's really tempting to say NADP or NADPH. I get that. Try to remember that NADP is only in the process that starts with a P, which would be photosynthesis. So NADP is in photosynthesis, right? But NAD is in cell resp. Okay, cool. All right, so then there's a reaction here that links glycolysis in the cytoplasm with the Krebs cycle in the matrix of the mitochondrion. And what fancy name do we give this reaction that links these two?
Link reaction.
The link reaction, yep. Okay. The link reaction converts 2-pyruvate into what are called acetyl coenzyme A, but nobody says acetyl coenzyme A. We don't say the coenzyme part. We just say acetyl-CoA. Okay. What happens is this is a 2-carbon compound, and the 2 carbons by themselves are so loaded with high energy electrons, they're unstable. Okay. So a cofactor from vitamins, as a matter of fact, is bonded to that 2-carbon compound to stabilize it. That acetyl group, that 2-carbon acetyl group is unstable because of all of the high energy electrons. So to the acetyl group, you bond a coenzyme A, thus acetyl-CoA. Ta-da. All right, and there's two of those, of course, and each of them has 2 carbons. So you have 4 carbons, but you started with how many?
6.
6. You have 4, you started with 6. So you know what comes out of the link reaction.
Carbon dioxide.
2 carbon dioxide. So a third of the CO2 that you're breathing out right now comes from the link reaction. Okay. Now, at the same time, some high energy electrons are captured by good old NADH. And you say, why doesn't Campbell list it? And I go, that's a really good question. I don't know. So we add it, because usually Campbell's diagrams are, they're okay. These diagrams are awesome. But for some reason, they don't include link. Okay, fine. But we're going to add it. Cool. So now the 2 carbon acetyl-CoA enters the Krebs cycle. A whole bunch of chemistry that we'll talk about tomorrow, maybe Tuesday. It's fine. You'll be fine. Let's see, Krebs. So then we have 2 electron carriers here. What are the 2 electron carriers here with their electrons?
FADH2.
FADH2 and? Cool. This also makes ATP, only a couple, just 2. And then the remaining portion of the CO2 comes out here. How many? 4 of them per glucose. So 2/3 of the CO2 that you breathe out comes from decarboxylation during Krebs. 1/3 of the CO2 that you breathe out is from the decarboxylation during link. All of the CO2 that you breathe out, assuming you're not doing anaerobic respiration, assuming you're not exercising vigorously, comes from link and Krebs. Super cool. All right, then ETC. I love how simple this is. It's just arrow, right? There might be a little chemistry here, just a little bit, right? So then notice how the starburst is bigger here. It should be a lot bigger because this would be 32. Whoops, that should be a 3. Okay, so do I change that to a 3 or do I squeeze a 3 over here? What do you want?
Change it to 3.
Change it to a 3, you got it. Not the prettiest 32 I've ever written in the world, but we can do it. There we go. So per glucose, right, you get a total of 36 ATP. The numbers of ATP produced per cell are in the trillions. The numbers here are astronomical, let alone when you start getting into neurons. Neurons use triple, if not quadruple, the ATP of all other cells in your body, even muscle cells. The majority of the food that you eat goes to your brain, right? Like 30 to 40% of the energy that you eat goes to power this little gelatinous mass. Okay, so when we talk about 36 ATP, this is literally the drop in the ocean of how much ATP is produced by your cells in your body every day, let alone the 6 billion people, let alone all the organs. It's just the numbers are just mind-boggling. Okay, so when 1 glucose comes in, here, 36 ATP. Your mileage may vary depending on your professor, but most of us, 36. Okay. One other item we need to add, speaking of breathing out CO2, what do you have to breathe in or you'll die?
Oxygen.
Oxygen, you bet. O2. And then that oxygen ends up in the water of your urine. So the next time you're in the bathroom, tinkle, 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 this is what you'll think about. You're like, really, man, do you have to do that? Yep, you bet. Because that water contains the low energy electrons that are the byproduct of your electron transport chain. And one day, photosynthetic organism, a plant, We'll reabsorb that water, and here we go again. It's all the same stuff, just cycle, cycle, cycle. It's the same oxygen, the same carbon, the same water, the same electrons that the dinosaurs metabolized 600-800 million years ago. It's all the same. It just cycles. Any questions about this before we leave it? I don't want you to be intimidated about this at all. It's a flow, step by step. Biochemistry should always be super straightforward. Comfortable?
Yeah.
Good. All right. So we're going to jump in there. So we need to talk about glucose to pyruvate, producing NADH and ATP. We need to talk about glucose to pyruvate, producing NADH and ATP. It's all the same. We need to label this first. So up top, glycolysis. Where does it happen in the cell?
In the cytoplasm.
In the cytoplasm. Boom. Then we need to go right across here. Here. Because there are different phases to glycolysis. There's the investment and the payoff. I'll explain. So this is glucose. 6 carbons, okay? Now, there has to be energy invested by the cell in order to start this process, okay? So 2, let's get a big old 2 in here. 2 ATP are invested, okay? And that, of course, leads to ADP plus 2 inorganic phosphate. Make the chemists happy with the PI. That's what that little circle is there. Fine. Okay, so 2 ATP are invested. So this is commonly called the energy investment phase. Have to put something in order for you to get more out, to get a payoff at the end. Cool. So then what happens is by a process called substrate level phosphorylation. Again, I get to just write that on the arrow once again. Just this chemistry is not complicated. There's just so much of it. Okay, I am in no way talking down to you like you can't handle it. That's not the case at all. Okay, I just don't have enough time, right? You will hit this in college. It's not difficult. It's just more detailed. There's always more, all right? So there's a process called substrate level phosphorylation where phosphates are directly added to the substrate. There's no ATP synthase. There's no chemiosmosis. It is directly onto the substrate. What happens though is that 4 ADP and 4 inorganic phosphates are able to be harvested. And so that ultimately, by substrate level phosphorylation, produces 4 ATP. Okay. So you put 2 in, but you get 4 out. Okay. So this step is called the energy payoff phase. Investment, payoff. Cool. Then, if we're going to have NADH here, right, this is going to be NADH, then what has to come in to pick up the electrons and form NADH? What goes here? NAD, totally agree. As a matter of fact, there's 2 of them. 2 NAD plus 2 associated protons will ultimately make, where's my marker? We'll make 2 NADH. All right. So what happens to your substrate if the electrons are taken out of your substrate and transferred to the NAD? What do we have to put on that arrow right there?
Oxidation.
You bet, oxidation. So your substrate is oxidized, right? Cellular respiration is an oxidative process. Your substrate is oxidized, the high energy electrons are pulled out. Those high energy electrons are then bonded to these protons and the NAD. Therefore, what goes on that arrow there as the NAD accepts the electrons?
Reduction.
That would be reduction. Nice oxidation reduction reaction. And then your substrate is produced, which would also be what is the product of glycolysis? Well, it was 2 ATP, 2 NADH, and
Pyruvate.
You betcha, 2 pyruvate. Pyruvate. How many carbons per pyruvate? 3. You haven't lost any carbon here at all. There's no decarboxylation in glycolysis at all. Okay. So let me back that out just a little bit. The products are called the net products. And that net is important. Have you heard of gross income versus net income? You heard people talk about these terms?
Yeah.
Okay. Do you know the difference between gross pay and net pay, or gross income and net income? What's the difference? And if you don't know, that's totally fine. Actually,
Net is overall.
Overall, what you get after taxes. Okay. So gross is what you're paid by your employer. Net is what you actually get after you hand it out to everybody else, after you pay your taxes. Okay. So I have a gross salary, right, which is what the school district pays me. Then I have the net income, my net salary, which is what I actually get to put in a bank account and, you know, pay for living indoors and food. All right. So here you have a net gain. Okay. And these are all 2s. All of them. What are the net products, or what is the net gain of pyruvate? Because all products of glycolysis are 2. What are they? 2 what?
ATP.
2 ATP.
NADH.
NADH.
Pyruvate.
Pyruvate. And you say, hold it, man, or you can't count. You say 2 here, but it's 4 here. Why, if 4 come out, do we say the net gain is only 2?
We invested 2.
Yeah, you had to invest 2 to start with. Okay, this is the same thing of financial investments. If you put $10,000 into an account and you wait 5, 6, 7, 8 years, right? And you get $16,000, $17,000, $18,000 back, and you go, oh wow, I just made $16,000. And you go, no, I didn't. Right? You made $6,000, right? Because you can't count your $10,000 that you originally invested. Okay, so you invest 2, you get 2 out. So your gross gain is 4, but your net is only 2. Comfortable? You okay?
Yep.
All right. So notice the color coordination by Campbell, right? This kind of blue aqua color here matches the blue aqua color here. They're matched. Okay. So now what we're going to do is we're going to talk about the link reaction there. Okay. And the colors here are equally as important. Stick that over there. So he has kind of a darker yellow here and then a very pale beige here, which is what they're showing you on 910. Come here. This is what they're showing you here. Okay. How big can I make this? Now we can do this. Okay. So this here is the inside of the mitochondrion here. So this is the mitochondrion here. This big old orange line, I wish he had put a big old orange line right here. This is not the plasma membrane. This membrane right here is this line right here. That is the mitochondrial membrane, the outer mitochondrial membrane, okay? And then this beige out here is the beige out here. This is the cytoplasm out here. So what we're doing in this handout is we're zooming in on that arrow right there, is that arrow right there. This arrow and this arrow are the same. Does that make sense?
Yes.
Okay. I should keep that just for giggles. So let's be sure that you are comfortable where we are. I don't need that handout. No matter how much desk space I have, I always want more. Always. It's like storage. Same thing. So that membrane right there is called the outer mitochondrial membrane. This is the outer, because just like the thylakoid inside of the chloroplast is double membraned. The mitochondrion, double membrane. This is the outer mitochondrial membrane. Like such. Well, if that's the outer mitochondrial membrane, then what is this fluid out here?
Cytoplasm.
Yeah. Cytoplasm. Link reaction starts outside of the mitochondrion. It should be starts and ends inside of the mitochondrion. Oh, squeeze. There you go. Thanks, Kenny. Okay. Starts on the outside, ends on the inside. So what we're looking at here is the product at the end of glycolysis. What we have here is a 3-carbon compound. What is that 3-carbon compound?
Pyruvate.
Excellent. Pyruvate. With its 3 carbons. Okay. If you want to know, there's your shuttle, protein called malate, therefore it's the malate shuttle. So the shuttle is a it's a channel protein that transports pyruvate into the mitochondria. You then rip off 1 carbon. So what goes in that circle there?
Carbon dioxide.
Carbon dioxide. And that carbon dioxide is removed by what process?
Decarboxylation.
Sweet. There's the box, just like the box we had over here. It's even the same color. It's almost like Campbell uses color to communicate with the right-hand side of your brain, right? That nice kind of, what is that, kind of a burnt orange-ish color, okay? Same there. So what carrier is there? What carrier comes out of the link reaction?
NADH.
NADH. Same thing here. We're in the link. There's your NADH. We just talked about your CO2. So this is NADH. So if NADH is here, what do we have to put here?
NAD.
NAD. What else can we add to that? Arrow here and the arrow right there. You can add your substrate and your NAD. Your substrate is oxidized, question mark. Oxidation, or you can write oxidized, same thing. So you have the oxidation of your substrate, and then you have the reduction of your electron carrier. See, this is why we can rip through cell resp pretty quickly, because you're familiar with carboxylation to bond high energy electrons to the carbon. What happens when you rip the carbon dioxide out? Decarboxylation. The movement of electrons, oxidation, reduction. You're familiar with the electron carriers. We can actually go pretty fast. So you had 3 carbons, high-tech math. 3 minus 1 leaves you with 2. What is that 2-carbon compound? Acetyl-CoA, nice. Question mark again. Nicely done. Acetyl-coenzyme A. Okay. And you're like, hold it. What's this sulfur and this coenzyme group, and what's with this swirly bond? This is a temporary bond. This is a very, very unstable acetyl group. Why? Because of all the electrons that are being carried right in there. It's just packed full of energy. So the coenzyme is actually a product of different vitamins. This is why you need to take your vitamins, and how vitamins help your metabolism be more efficient, in that it stabilizes with this highly unstable, but it's more stable than it was. This is a temporary bond here. So this is what coenzyme A really looks like. And so thus this is your sulfur bonded to a coenzyme. And we don't put sulfur into the name. I caught that one. So this is just referred to as coenzyme A. In college, there are two kinds of nutrition classes. There are nutrition classes that talk about nutrition in general, and then after you take some biochemistry, you then go take biochemical-based nutrition classes. Holy smokes. That's where you really start putting things together. It's awesome. Biochem for bio majors puts everything together. Here's the chemistry, here's the physics, here's the biology. Bang, it all fits together. And how do we measure all of this biochemistry? With calculus. And you're like, oh, right? But sometimes as a bio major, you're trudging through the math classes and the physics and the inorganic chemistry and the organic chemistry. And you're like, what's with all these classes? And then you hit biochem, and it's ta-da, and it's this beautiful aha moment, okay? Or you get people into biochemistry and they go, oh, I thought, boy, I should have studied more back when I was in chemistry and calculus and physics, okay? So when you're in your lower level science class, okay, and you're in your entry level class, just trust the process. They're going to put all these foundational classes, and then the ultimate goal for the bio majors is your physiology class and your biochemistry class. That's the other aha class, is you get into biochem and it links all the classes together. And then once you have your biochemical foundation, then you start talking about the biochemistry in the different organ systems. And you get into the biochemistry of your brain, and the biochemistry of your liver, and the biochemistry of your stomach and muscle. And then you're really rolling. And you're like, oh, I get this. It's not about memorization. It's conceptual understanding. And ta-da. And then someone says, what's messenger RNA? And you decide where to start. Really, yeah. I was sitting on this past Saturday. We've not seen my mom. I hadn't seen my mom for 6 months in person. It was really nice to see her. And during the conversation, she and her 3 80-year-old friends turned to me and said, okay, we heard this vaccine really isn't based on viruses. That doesn't have any viruses in it at all. It has something in it called messenger RNA. Yeah, and they said, what's that? Okay. How do you teach that, right? I mean, I did it, and from what I understand, it turned out pretty well. But it was I was laughing that, usually for work I'm introducing messenger RNA to 14-year-olds, right? Going, okay, messenger RNA, here we go. And I had never anticipated introducing messenger RNA to 80-year-olds. It was really cool, but it was a really good question because they wanted to know. It was awesome, super cool.